Okay. Hello, everyone. My name's Cahill. I'm a PhD student at the Uni of Manchester, working with AI for protein engineering of monoterpene synthesis. And I'm also one of the co-MDs from the Manchester chapter of Nucleate UK. I'd like to welcome you all to our intro session to AI and protein design. This is the first part of our AI and biotech initiative in partnership with Bits and Bio. We also have an AI and protein design panel discussion, and we're creating for anyone interested in getting into this field. Um, this effort is to make the field more accessible and has been running in tandem with the Nucleator Activator Program. Um, Personally, I find this field really exciting because I believe it can have a massive positive impact on the future across a wide range of fields, and it's continually had massive leaps forward in the past 5 years. Today we have Paul Kim to talk about these advancements and give a background on AI and protein design. Um, Paul has experience working at the Bayer and the New York Structural Biology Center as an ML researcher. He's now a second-year graduate student in the University of Washington Baker Lab, which is one of the most prominent labs for protein design in the world, and they've actually developed some of the tools that will be discussed in this talk. So, Paul, do you want to quickly what you're working on, what you brought you into the field, and then teach us a bit about protein design?
Yeah, thanks for the intro and happy to be here. Really excited by what Nucleate is doing and happy to be able to contribute. Yeah, so I started off kind of in the intersection of biology and machine learning, particularly in small molecule drug discovery. And then I got more interested in working with proteins. And I think one thing that's really nice about proteins is the tight linking that you can have between the wet lab and the dry lab. Since generation of new protein sequences is pretty easy in the real world, relative to making like a new arbitrary small molecule, at least. And so that's what kind of drew me toward this field, especially to the Baker Lab. And so my current project is to work on improving our diffusion models, our structure generation models, which I'll talk about today, for the development of de novo DNA binders, so de novo proteins that can bind to DNA and make interesting interactions with DNA. So that's like we have a little subgroup of people that work on this, and I'm working on more of the computational methods in that space. But yeah, I guess without further ado, I'll go ahead and share my screen here. Okay, great. Yeah, so this is going to be just like a quick intro, about 45 minutes, 1 hour intro to the de novo design space. This is going to be a slightly longer and more formalized version of a chalk talk that I do with new rotation students in the lab every quarter when they join, just introducing them broad strokes to the methods. And yeah, this will be very kind of biased to the work that is done here at the Baker Lab because it's hard to know everything that's going on in the rest of the world. So if I leave important papers out, I apologize in advance. But I just want this to be out there as a resource for people who are trying to get into the field. So I think the first question that we need to answer is like, why do we want to do de novo design? Especially when other protein engineering methods exist, especially stuff like directed evolution. So I think the main draw here is that for de novo design, there's no native starting point required. So in directed evolution, you usually start with some protein that has binding or activity that you're interested in, and you need that starting point. You then develop an assay that will find proteins that have higher and higher activity or higher and higher binding, and then you repeatedly cause mutations to the protein, assay them, take the stuff that had the best performance, the highest activity, and mutate those again, and so on and so forth. So this always requires some kind of starting point, usually some kind of native protein that's able to, at a very low level, carry out the function that you're interested in. With de novo design, you don't need any kind of native starting point. And so this, I think, lets us tackle a much broader range of problems and a lot of interesting problems that nature hasn't really designed a solution for. The other thing, I think, is that there is a potential for greater efficiency. So to do kind of like an apples to apples comparison here, you've got to think about the amount of computational work it takes to get some kind of, let's say, a protein binder to a small molecule compared to the amount of work that it would take to do that with directed evolution. And here there's a concept called kind of a hit rate that's pretty important, and that's sort of the fraction of designs that you make in silico in the computer that will succeed in the wet lab. And so at the moment, depending on the type of problem you're doing, the hit rate varies somewhere between, below 0.1% to upwards of 10%. And in the cases where we can get the hit rate really high, that means that we can save a lot of wet lab work relative to directed evolution because we do a lot of the hard work in silico on the computer, which is, orders of magnitude cheaper. And then with the right filters and the right methods, we can achieve really high hit rate and therefore really good efficiency. And I think, directed evolution is fundamentally bounded by the sort of wet lab process that it requires, whereas our computational methods are getting better and better every year. And so the hope is that we can really improve on these computational methods and get very good efficiency for tackling problems that you could also tackle with directed evolution. And then I think, besides just designing for function or for activity, there's a lot of interesting things that you can just do in the structure space that are much harder to do with directed evolution. For example, designing of scaffolds and structures like these on the right. These are protein nanocages that have different types of symmetries made up of either monomers or dimers. And they form these interesting structures that then you can use to deliver drugs, or you can stick viral antigens on the outsides and turn this into a vaccine. So there's a lot of room for interesting structures that you can make with de novo design that, this type of assay doesn't really exist. So it would be really hard to tackle this with a directed evolution campaign. So a lot of the things that we do in de novo design are actually built off of our understanding about protein structures that comes from the protein folding problem. And I think I want to first do a little detour into talking about that so that we kind of have an understanding of what type of information we've had to build off of in order to do de novo design. So first, the protein structure prediction problem. This has been a long-standing problem in computer science. The question is basically, given a protein sequence, can we predict the 3D structure that protein will fold into? And this has been going. There's been a competition for this since 1994 called CASP, where people submit servers or submit models and methods that are then tested on a held out set of recently crystallized or structure determined proteins, and the performance of those models is then evaluated. So a lot of the work that we do is built off of the structures that were deposited in the PDB, which is a large database of protein structures. These can be either crystal structures or cryo-EM structures, NMR structures that were resolved at some point in the past. And people have been submitting structures to the PDB for a very long time. This gives us very good training data for our machine learning methods. But when CASP first started, and for the first maybe 15, almost 20 years of CASP, the methods used were not deep learning based and were predominantly based on energy-based methods, substructure threading, and homology. We're not going to go into too much detail here, but basically, a homology model, you have some query sequence of interest, and you look for sequences of similar proteins for which you have a 3D structure already. And based on these, they're quote unquote homologs, you get some kind of starting point for what you think the overall protein fold is going to look like. And then you make some refinements here and there, or maybe you combine different templates from different sequences and try to piece together like a 3D structure. So that's one of the ways that people did protein structure prediction in the pre-deep learning world. And one thing that we kind of find is that secondary structure, so whether or not the protein is making an alpha helix or a beta sheet, or is kind of in a loopy unstructured region at this sequence location, secondary structure is relatively tractable, using methods like substructure threading, where you can just infer from other structures and other sequences that, this region is some kind of alpha helix. But tertiary structure, so how those secondary structure elements come together and make long distance contacts, that problem is much harder to solve using these pre-existing methods. So a really key idea then to try to get at these long distance interactions that will tell us how the secondary structure elements fit together in order to form the tertiary structure of the protein is this idea of evolutionary couplings. And so this is not necessarily a deep learning based method, but the modern deep learning methods take advantage of this information. Basically, what's happening is if you can get an MSA, so a multiple sequence alignment, this is a way of finding other sequences that are very similar to your query sequence in some massive data set, like in UniRef or in UniRef. So these tools were built and were used for stuff like homology modeling, and it turns out that they're also very useful for structure prediction using deep learning. But basically, when you get this kind of MSA, you can see that there are certain long distance, so sequence positions that are far apart in sequence space that have evolutionary couplings in how they are, in how those residues evolve over different sequences. And so what I mean here is that if you look at these two positions, every time this residue here mutates, it's met with a corresponding mutation at this other residue. And this kind of tells us that these two residues then must be linked somehow, must be important for the overall fold of the protein, even though they don't look like they'd be touching in the sequence space. And so this is a way of kind of inferring where the 3D contacts are that might be far away in sequence space, but are close together in structure space. And this makes sense, intuitively, if you can imagine you have a polar interaction that's forming like a bridge between two secondary structure elements. If you mutate that one of those polar residues into a non-polar residue, it's going to break apart your protein. And so this type of information can help you to figure out then where the 3D interactions that combined with the secondary structure information you can get from the sequence, you can start to really piece together what the protein's overall structure is going to be. And so in 2019, I believe, AlphaFold came out, and then it was subsequently followed up by AlphaFold 2. And these were massive leaps in the field. And these made very heavy use of these evolutionary couplings information. In particular, so above is kind of a schematic of AlphaFold 1. You input a sequence and an MSA, and now instead of kind of using a human to figure out, okay, here are the residue positions that appear to be evolutionarily coupled, and so we can say there's some 3D contact happening here. They just use a deep neural network to reason over the multiple sequence alignment and find the evolutionary relationships itself. They use that to then build a distance matrix that represents the distance in 3D structure of every pair of amino acids in the protein, and then they seed a structure with that distance matrix and do some kind of gradient descent in order to find a better protein. And this was kind of the first big leap in the field in many, many years. And then AlphaFold 2 came out, where the main difference here is that they did a little bit different type of deep learning in the middle section, where they're reasoning over the distance and the pair representations. And then instead of using a potential and doing gradient descent in order to find the best protein structure given a distance matrix, they just use a neural network to do this. And so the addition of the structure module basically really improves the ability of the model to actually represent the final prediction of the protein. And so this was very effective in getting really good structure predictions. But in general, these are underpinned by the evolutionary couplings information that you can find in the MSA. And so AlphaFold 2 was a enormous advance in the field. Just to kind of give a sense for this, on CASP 14, so the 14th edition of the CASP protein structure prediction contest, AlphaFold basically beat every other method. So, on the x-axis, these are all different types of proteins that were in the holdout set for the competition. And AlphaFold 2 just does better on basically all of them. And today, we live in this post-AlphaFold world where in the most recent CASP, CASP 15, all of the best methods were actually some type of improvement or hack on top of AlphaFold. And so in particular, one of the best ways that you can improve performance with AlphaFold is to be clever in how you pick what sequences go into the MSA. And all of these methods were basically different ways of doing that to try to find the most relevant sequences that give you the best evolutionary information to make your best protein 3D structure predictions. Yeah, and so a lot of our methods are kind of built off this basic paradigm of protein structure prediction. And in particular, we have tools like Rosetta Fold diffusion, which I'll talk about later, which are actually explicitly fine-tuned off of Rosetta Fold, which is a structure prediction method very similar to AlphaFold. But now moving on to the protein design pipeline. We basically split the problem up into, you could say, 3 or 4 pieces. So the first part is just backbone design. So independent of sequence, we just want to find protein backbones. So these are the C alpha, C beta, and nitrogen atoms that form the protein backbone, or C alpha, oxygen, and nitrogen atoms that form the protein backbone. And we want to generate interesting backbones that might be able to do a function that we're interested in. Let's say that's bind to a protein, or maybe bind to itself and oligomerize in some interesting symmetric way. But that's usually step one, is just to make protein backbones. The next step is sequence design. So now, given protein backbones, the question is, can we find sequences that will fold into this protein backbone that we're interested in? And so this is kind of called the inverse folding problem. Basically, given a protein 3D structure, give me sequences that might fold to this structure. And then we might do some steps of redesign, which I'll talk about what this means. But basically, there might be some intermediate steps of trying to improve the backbones and the sequences that we generate. And then the final step is to do refolding, or collect other types of metrics. And so refolding here is where you generated a protein structure, you generated a protein backbone, you fit a sequence to that backbone, and now you take that sequence and put it into a protein structure prediction method like AlphaFold and get a predicted structure for this sequence. And then you compare the structure that the model predicted with the structure that you were originally trying to design. And that gives you a little bit of internal consistency to try to understand whether or not this sequence is going to work to design this structure, and also whether or not this structure is designable at all. This is a bit of a hacky solution, unfortunately. It's kind of a proxy for the real information, which is actually trying out the protein in the wet lab. But absent better computational filtering methods, this is actually a good way to do things. And we find that by filtering based on these refolding metrics, we're able to enrich our libraries for higher quality proteins that are more likely to be soluble and more likely to perform the function of interest. So I'm going to switch gears to my iPad here so that we can draw out some of the methods. So the first method I'll talk about here is called RF diffusion. And this is a method for generating new protein structures, new protein backbones. And so this is built off of RosettaFold, the structure prediction algorithm. And in particular, it's a fine tuning of a structure prediction algorithm. So basically, we trained a model on protein structure prediction, and then we took the weights of that model and fine tuned it on this new generative task. So to basically introduce diffusion, instead of talking about proteins, to keep things a little bit simpler, we're going to start off by talking about images. So let's say that we want to generate images of faces. And by images, what I mean here is, let's say, 64 by 64 grid of pixels, where each pixel is some value between 0 and 1 that represents the intensity. And so, everywhere around here, it's going to be zeros. And then, in the face itself, there's going to be some like 0.5, 0.8, whatever, some like larger numbers where it's darker. And for simplicity, let's just keep it a black and white image. So I don't know how to just like sample images like this. I don't know a way that I don't know some kind of mathematical function that I can just run in order to get images that look like images of faces. Okay. However, I do know how to produce a mathematical function that will give me images of totally random static. So this could be kind of like a TV screen that's kind of static, and it's just like black and white, and it's totally noisy. I know how to get images like that. All I need to do is say, get a random number generator and parameterize it as a Gaussian distribution, and then sample from that Gaussian distribution and let each value that I sample represent the pixel intensity at a given pixel, and I make each pixel independent. So I just sample, 64 times 64 times to get this like random static image. So we have some distribution of, noise, and we don't know how to sample from it, or we know how to sample from it. And then we have some distribution of more structured data that we're interested in that we don't know how to sample from. And so the trick with diffusion is basically learning a way to bridge these distributions, learning a way that then I can sample a fully random noised image and then run a model to transform that into a more structured image of a face. Okay, so how do we do this? The first thing I want to do is define two quick terms. We have training. Oh, oops, wrong. Here we go. Training. So basically at training time, what this means is we're just trying to train the weights of the model. So we want to get a model that's able to do some kind of interesting task, in this case, generate proteins. And then in addition to that, we have a concept called inference. And inference just means now we have a model that's been trained, and our goal is to use this model to, in this case, generate the images that we're interested in. So now this is the part where we actually use the model that we've trained. So our goal then is to train a model that's able to, at inference time, invert this problem where then we can sample from pure noise and generate a image of a face. So the way that we actually do this is kind of conceptually actually relatively simple. We start with a data set of faces. We have this data set of faces that we're going to use as our ground truth. And what we do is we corrupt this image by various amounts. So if there's kind of like a continuum between full structure on this side and full noise on this side, then in between, you can have images that are more or less structured based on how much noise you've input. And so you can imagine there can be very little noise. And so we have the picture of a face, and it's just slightly corrupted here and there with a bit of noise. And this is maybe something that's pretty easy for the model to figure out, like, what is the ground truth image that this noised image belongs to? But, we could also imagine that we corrupt the image much more. And now, the image is mostly like blurry and hard, but you can kind of make out maybe that there's like a face there. And so then the task of the model is to figure out what image this originally came from. Now, as like a machine learning note, there's another way to parameterize this, which is to parameterize the steps in between two noise states. But just for RF diffusion's purposes, at least, we found that it works better to parameterize it this way, where we're trying to predict the ground truth structure that the protein came from. So in this case, predict the ground truth image that some noised image was generated from. And so we do this for very, very many time steps and for very, very many faces. And then the model basically learns how to invert the noise process, and thereby learns the mapping from a noised image to a ground truth image. And so then at inference time, what we do is we just sample from a fully noised distribution, and we run the model that we've now trained to predict the ground truth. And maybe in the beginning, it's not really going to know what it is, so it's going to kind of predict something kind of fuzzy, let's say. And then we take this image and we re-noise it to one time step forward from the initial fully noised state. So now it'll be, again, noisy, but there'll be some kind of structure from the previous prediction in there. And the model then takes this and again predicts the ground truth. And now there'll be a little more structure in the image. So it'll kind of be able to predict it a little bit better, let's say. And, but it'll still be, noisy and tough to figure out. But then this image gets noised again. And we repeat this process over and over again, gradually reducing the amount of noise that we add until at the very end, we hope that the model will be able to produce an image that looks like an image of a face. So that's basically how you would make the denoising diffusion model in the domain of images of faces. What we do is we translate that into the domain of proteins. And so for proteins, basically we have some protein 3D structure, ground truth structure, let's say. And this is like the actual 3D structure. And to noise it, we basically move all of the atoms in the protein, and we, actually we deal with these backbone frames, but basically you can think of it as moving all the atoms around and rotating them in random orientations. And so then our noised example is going to be a whole bunch of random protein backbone atoms kind of in space. And then the task of the model is to figure out what protein this came from. And so then we train the model on various levels of noise where, at different points, there might be more or less structure available that came from the ground truth versus like a lot more noise, but a little bit of structure, let's say, and then full noise here. And so the model is just trained to basically reverse these noised protein structures and produce actual realistic looking structures of proteins. And then at inference time, we run it the same way, where we then now we sample from actual just full noised, Gaussian noised protein atoms, and we task the model with iteratively figuring out what is the ground truth protein that this noise would produce. So yeah, that's basically RF diffusion. It's a way of generating new proteins that look like they came from our data set of interest of protein structures that we can sample from by starting from pure noise. So this is how we generate protein backbones. But then the next stage is to fit sequences to these backbones. And for this, we use a method called protein MPNN. And what protein MPNN does is basically it starts with a protein 3D structure, and these are really bad protein schematics. Let's try a slightly better one. Let's actually make it a little bit more protein-like. Okay. So you start with some kind of protein's 3D structure, and what it does is basically it goes through all of the local regions of the protein, and it encodes a 3D representation of all of those local regions into sort of a graph neural network. And so this graph neural network basically processes the distances between atoms in the backbone of this protein that we're trying to find a sequence for. And then it has a graph neural network encoder, and then it fits it into some kind of latent space, which is just a set of numbers basically that represents the structure of the protein in some higher dimensional space. And so then this latent space is an encoding of this 3D structure that the neural network understands. And so then it goes into a decoder, which reads from this latent space that was encoded and predicts the amino acid sequence. And so then it predicts, it predicts an amino acid sequence that corresponds to this input structure. And so we can do this, and we can actually run the decoder in different orders of what the order of the sequence we decode is, and that will give us slightly different predictions for the sequence. And this means that we can actually run this many different times to get many slightly different sequences. So so we started with nothing, and we generated a protein backbone. Then we fit several sequences to this backbone. The next step is to then take these sequences and fold them. So then we take the sequence that we generated, whatever, and we feed it into AlphaFold. And in particular, actually, when we do this for de novo design, we usually don't include the MSA information. And so in this case now, the model has to go off of just what it knows about how to fold single sequences, which actually is quite a lot because it's gotten a lot of information from MSAs, and that's taught it something also about, just for a lone single sequence, what is it likely to fold into. And so then AlphaFold gives us a protein 3D structure prediction. And what we do is we compare that 3D structure prediction with the design model that we originally wanted to generate. In particular, we look at what's called the C-alpha RMSD, which is basically the distance between corresponding alpha carbons in the protein that we generated versus the protein that we wanted to originally design. And so this kind of gives us like I said, a bit of internal consistency that can inform us whether or not the sequence that we picked is a good sequence that will fold into the structure that we want, and also give us a bit of information as to whether this structure is designable at all in the first place. What C-alpha RMSD is good kind of varies by the task that you're trying to do, but in general, we're looking for a C-alpha RMSD of below 2 angstroms, and ideally, below 1.5 angstroms. These are kind of arbitrary cutoffs, but these have been validated in the real world. So basically, if we filter by a C alpha RMSD cutoff of below 2 angstroms on average, then we're more likely to get sequences that fold to the desired shape in the real world. So that's the basic pipeline. We use RF diffusion, and we generate protein backbones. And then we use MPNN, and we generate sequences. And then we take those sequences and feed them into AlphaFold or some other protein structure prediction algorithm, and we get structures again. And then we compare those structures to the design model, and that gives us one of our most important metrics for figuring out whether or not this structure is good, or whether or not the sequence and the structure is good. But of course, this is the most basic abstract version of this pipeline. There's a lot of other steps that we can do. One thing in particular is we can do filtering before we get to AlphaFold. So the thing is that running AlphaFold is computationally intensive because it's running this large deep learning protein structure prediction model. And so ideally, we want to then sort of reduce the amount of things that go into the next step by looking at the sequences and coming up with looking at the sequences or looking at the sequence now just naively threaded onto the structure that we've originally designed, and produce computational metrics, sometimes using the energy algorithm, physics-based energy modeling algorithm, Rosetta. And this can tell us like what types of hydrogen bonds our structure and our sequence might be making. How likely is it to fold, or how likely is it to be soluble? All these different types of sort of like statistics that we can generate from the sequence before passing it into AlphaFold, we like to do. We might also try to save computation at the MPNN level, although MPNN is very cheap to run. MPNN, you can just run it on a single CPU. It doesn't require any GPU, but you can do some filtering at the structure level where you can say, oh, I want structures of a certain type of topology, and if they don't have the right shape, then I'm not even going to bother trying to fit a sequence to them. So we can do a lot of different types of filters. We can also do a lot of different ways to diversify our method, our generations in silico. And so by diversify, what I mean is something as simple as we take something that we've produced with AlphaFold and we like it, and we say, okay, let's try to make more stuff that's like this. And so one way we can do this is we can just going up to the RF diffusion inference procedure here, where we start where now we start from noise and we produce a structure. Now, let's say we have some structure that we like that AlphaFold also said was like a good structure. Now we just diffuse it part way. So we add, not all the way to full noise, but we go just part way. And now, some information about our protein is left, but also some atoms have been moved around. And so this is like a good starting point. And so then starting from this now, if we run inference on this sort of partial diffusion example, then we're going to end up with a final structure. Then we're going to end up with like a final structure that probably is going to look, something kind of similar to it, but might have some slight deviation, let's say. And so this is basically one of the ways that we can diversify the designs that we generate by taking things that looked good via AlphaFold and then running the pipeline again, but not eliminating all of the information from the original structure and instead doing a partial diffusion. We can also do stuff like redesign with MPNN. We can just say, okay, I think that this structure looks really good, but maybe we can find new, better sequences that are better equipped to fold into this structure. And so we can basically go in all different types of ways in order to diversify our structures and not just have to run this pipeline linearly all the time. So yeah, that's kind of the chalk talk overview of these different methods and how we kind of put them together in the lab. I'm gonna now switch back to the slides here to just talk about some of the challenges and also kind of what we're able to do well right now. So I'll do that right now. So this is the pipeline, and using this pipeline, there are some things that we can do, at least in the lab, fairly easily. And by fairly easily, what I mean is that if someone has some goal in mind, some design project in mind that they're trying to accomplish, and it's one of these things, it's fairly consistent. It's highly likely that we're able to find a protein that does the function of interest. And so these things are stuff like making binders to other proteins, in particular nonpolar regions on other proteins. And so here we have like an example in green of a binder target, and via diffusion, we gradually denoise a fully noised structure into a new 3D structure that's likely to bind to this target protein. We're also able to do stuff like motif scaffolding, where we have some functional motif, let's say, that we want our final structure to have. And so then we provide this as context to the diffusion model, and we tell it, make a protein that will fit around this structure. And this is another thing that we can do fairly consistently. As you saw in the first slide, we can also make these multi-domain symmetric and pseudosymmetric complexes, and we can use those to generate interesting nanocage-like structures or even little molecular machines. We can also do some small molecule binding, especially to nonpolar small molecules. And we are also able to do some types of goal-directed sequence design. And this is a little bit off topic from the pure de novo design, protein design that I was talking about, but this is kind of a fun thing that you can do. Let's say there's some membrane protein that you'd like to characterize better. And the problem is that membrane proteins in general are hard to work with. We can actually make solubilized versions of membrane proteins that have the same 3D structure, but a different sequence that will let it localize to the cytoplasm instead of the plasma membrane of the protein. And so these are things that are fairly doable with the current set of tools. But that leaves a lot of challenges and open problems, both on the design side and the computation side. And some of these problems kind of loop into each other. And so the first big question is how to do better polar interaction design, both for protein-protein interaction and protein-small molecule interaction. So to make this more clear, we have this small molecule here, and it has these nonpolar regions, and making some kind of shape complementary region to this nonpolar region that will be able to make good van der Waals interactions here is something that we can do in the lab, and something that people are able to do with fairly high consistency. But trying to get actually the polar interactions where you need then the side chain of the amino acid in the protein that you're designing to point in the right direction and the right angle so that it creates this hydrogen bond or polar interaction to a polar part of our small molecule target or a polar part of our protein target is much more difficult. And this is because you need a higher level of precision, basically. For just the nonpolar region, you just need to basically match the shape in a kind of fuzzy way. Whereas for a polar region, you need a specific atom to be within like a very tight, 1 angstrom range for it to be able to make a good polar interaction. And getting that level of precision is just a lot harder. We're also trying to design against loopy regions or unstructured regions. So a lot of proteins are actually in nature don't fold into some low energy 3D structure and instead are naturally floppy and loopy in the cytoplasm. And a lot of these then have very interesting functions. This is something that we're not really able to design. And I think that's something where this structure-based approach might not work. And instead, you might want to look at just like a purely sequence-based approach to protein design. And then kind of tying into polar interaction design is enzyme design, where often, then we need to first be able to make a binder to some small molecule target that might have polar regions. And then a lot of enzymes operate on some kind of dynamic function where the actual structure of the protein shifts upon binding in order to catalyze a reaction. And that type of dynamics is something that also we have a lot of difficulty with. In particular, with dynamics, our protein structure prediction methods always have been designed to predict static 3D structures of usually crystallized versions of our protein, which means that they very much favor the lowest energy conformation of that protein, or at least a very low energy minimum of that protein. And in reality, proteins have higher energy states, higher conformational states, and are present in water in the cytoplasm and being jostled and moved around all the time, trying to actually design new proteins that can have interesting dynamic behavior is something that we're still working on in the lab. And then on the computational side, of course, we'd love to make all of our methods cheaper, basically. Diffusion and structure prediction folding are both GPU intensive activities. Depending on the length of your protein, diffusion on an A4000 GPU can take somewhere between a minute to 3 to 5 minutes to 20 minutes if you're trying to diffuse something really big. Structure prediction is also relatively expensive. It usually takes on the order of seconds to get a prediction of a protein that for a sequence that you've designed. But if you have very, very many sequences, in particular, if you've, let's say, done, 10,000, you've generated 10,000 structures with diffusion, and then you've run MPNN to get 100 sequences for each of your 10,000 designs, then you're going to end up with 1 million sequences that you need to pass into AlphaFold. That means that, even if it takes on the order of seconds per sequence, you're going to run into a lot of computational cost. Kind of adjacent to this is trying to make alternative or new filters that can be predictive of good designs without having to go all the way through the pipeline. And instead, maybe if you could figure out like what designs are going to be good, what designs are likely to fold well from just the structure, from just the backbone, or from just the sequence, those things would be very useful. And I think the last thing, this is a little more like a new methods development type question, but basically, if we can do joint sequence structure sampling, I think that would be very interesting as well. So basically, so far, we've always been doing structure sampling on its own, and then, so unconditioned, or maybe with some structure conditioning, and then we sample sequence conditioned on the structure. But really, we know that there is this deep relationship between the sequence and the structure such that we could imagine that we can sample both the sequence and the structure at the same time. And so this means that we specifically sample a structure and sequence, and the sequence that we sample is one that our model knows is going to fold into that structure. And so trying to do this would actually probably improve our ability to design polar interactions and stuff like enzymes, because often then you can, if you know that you want this exact structure while you're trying to make a sequence for that structure at the same time, then you can know that, here there needs to be a polar residue. And so then we need to put the structure around it that can properly scaffold this polar residue so it makes an interaction or something like that. So having your structure design and your sequence design be aware of each other, and therefore doing this in a joint way, will probably help with that level of precision. And so this is one of kind of the major open questions and open challenges in the field. So yeah, of course, there are lots of other problems and questions that people are interested in. But to move on now, just going to give a quick overview of some of the major papers that I wanted to highlight. And we'll have the links to these papers in the resources that we'll send out, but also, you can just Google search for all of these. There's a bunch of different protein structure prediction methods now: AlphaFold 2, Rosetta Fold, ESM Fold, and OpenFold Colab Fold. Most of them pretty much use the same general framework of starting from a sequence. You take an MSA, and then you use that to predict some kind of 2D representation, and then you predict the 2D contact representation, and then you predict the 3D representation, the 3D structure of the protein. There are some other methods that I didn't mention here, but so ESM Fold and Omega Fold actually use a language model instead of an MSA, and so this they hope will make their models faster and able to work with sequences that don't have a lot of similarity to other sequences in the world. And then there's methods like RF diffusion and Chroma. So RF diffusion is a Baker Lab thing. Chroma was made by Generate Biosciences, a biotech startup in Boston. There's also NOS, which is made by Prescient Design, which is part of Genentech. These are all structure generative methods. Chroma also does some sequence design. And then we have protein MPNN, which is probably the go-to for sequence design. There are also some other methods for this inverse folding, given a structure to sequence problem. But I think protein MPNN is pretty easy to use and works quite well. And then just in the sort of sequence generation space outside of this like structure-based de novo design, there's methods like POET and ProGen and ESM. And these are all different ways of generating new sequences or doing conditional generation of sequence, kind of abstracting away the structure side of the equation. That's the extent of what I have, and I hope this is helpful. And good luck on your projects, and best of luck with getting into this field.
Is there anything final you want to say at the end, if you wanted to say a final word?
It's a real privilege to be able to work with Nucleate on this, and it's a lot of fun for me as well. I really enjoy disseminating this kind of information and making this more available to all the amazing scientists out there who will hopefully do wonderful things in protein design in the future.
Hi, it was brilliant. Thank you so much for coming along, Paul, and covering this exciting field. As I said before, we have a number of similar things we are putting together as part of the protein design and AI as part of Nucleate and AI Biotech Initiative, including a resource page, which Paul mentioned, and a panel with Paul and some other researchers in the field. Following along from that, we have in our biotech initiative, we'll be covering some other areas, including drug discovery, and automation, data management and regulatory compliance, and finally biomedical AI ethics and alignment. So if you're interested in them, follow along with the initiative. Okay, thank you.